All right. Hello, everyone. First, I'd like to thank the Michelle and team for organizing this great event. It's a great pleasure to be here. Very interesting. And well, if we think about imaging pipeline, so we start with a scientific question, scientific problem that we would like to address using imaging, various microscopy modalities. And within this, with our pipeline, we go through the different steps, starting with sample preparation, imaging, image processing. But finally, we end up to the segmentation, quantification, and potential deposition and data reusage. And these would be the topic that I would like to cover today in my kind of short talk. Well, if we talk about segmentation, the most natural thing that comes in mind these days is deep learning. And this plot on the left-hand side shows the utilization of, well, basically, how many times deep learning or convolutional neural networks you can find when you do analysis of PubMed database. You can see there is a very steep rise in these techniques, and which is very, very interesting because this happens within this biological society or biomedical research, which is a little bit conservative in some ways in order to apply these more heavily computational methods. And indeed, it's a bit tricky to start with because there are multiple things that a little bit slow down application of technologies. Nevertheless, it's very powerful, and that's why we really have to consider this. Florian already showed a little bit how it works. Basically, what we have the provide images and the labels or models, which we segment something that we want network to be trained to predict. And then it goes to the network of different architectures, whatever you prefer, and at the end, network compares whatever it generates with whatever we provided. And then when it's trained, we can apply it to new data, and thus we can actually predict significantly large amounts, like the big data sets, the full data sets, using only very small data for training. So for more than 10 years, we've been developing software for image segmentation. We started because we got the serial block-based system and we need to somehow work with the data. So there are many tools in there that I don't have time to go into details, but I just want to highlight that we have quite many different manual, semi-automatic, and also automatic tools with deep learning that allow you to train networks and then apply it to your data. And then we try to do it in a way that is like as easy to use as possible. You don't need to have any knowledge in computer science, or, well, it's good to have some ideas about deep learning. There are different, when you go into the software, you can select different architecture or different workflows. You can do 2D, 3D, 2.5D, different workflows. And within this workflow, we selected different architectures that work quite well on the volume EM data sets. In general, again, I don't want to go that much in details. We go through these different steps when we can define the folders, we can define the different settings for the training of the networks. And finally, in the last step, we just go and predict the full data sets when the network is trained. It's important, the interesting thing that you just need only a few data if you want to train a network, especially in the volume EM, but it's important to augment the data. So you need to provide the network with a variety of options, and this is done through augmentations. And what we try to do, try to make this kind of design that you can specify the filters that you want to apply. You can specify the probability of each of this filter to be triggered. And then also you can specify what will be the variation. Let's say, for example, if you introduce the contrast jitter to make sure that the network is trained on the data, if it's brighter, dimmer, or has stronger or weaker contrast, you can apply this, and you also can see the result of this application, this augmentation, by just clicking this kind of the I button here on the right-hand side. When you hit train, then you start training. It's very interactive, so you can see sort of this loss function, which is the difference between the result what network predicts versus what you want it to do. And in general, we try to reach as close as possible to zero. Hopefully we want to reach zero, because I think in this case, potentially you kind of probably messing up something, and maybe network learn the patches instead of actually learning the features of your images. And then finally, when network's trained, we can go and evaluate this. And one of these evaluation results, what is important, that you really fine tune the network, you can extract these different metrics, performance metrics from your trained network, and see how well you go, and then potentially gives you possibilities to fine tune the model. So, well, okay, we have these networks, we have data, let's say, we have the technology to generate networks to predict the data, but how we can generate the training sets? It's been already mentioned a few times today about segment anything. So this is pretty cool stuff, which was released in April last year by Meta AI. where the idea was that I envisioned that with a single click, you segment the whole image in different objects and classes. Well, the problem for us that it was trained on normal images, natural images like dogs, cats, and it works really well for those. But for EM, it's sort of, well, it probably doesn't really work. But it works in a way more like an interactive approach where you do yourself like interactively click on something and that object gets segmented. And the reason for that, the features that we have in EM, in microscopy, basically the similar stuff as we have in normal images. We have the blobs, objects with a different contrast and colors and things like that. It's important that segment anything was updated couple of months ago and maybe a few weeks ago to version 2.1. And this is like way overperform the version 1. So it's way quicker and actually can segment things that the first version was not able to do. Here's an example still from the first version where I try to segment different objects from the different volume EM modalities. So there are trypanosomal profiles from serial localized SCM, mitochondria from tomography, lipid droplets from FIPSAM. And basically all of this can be done pretty much with a single click. So now let's say we generated this training set. It's very quick and easy to do. And now we want to train a network. Again, there was a previous talk which was very nice, already highlighted the application of various approaches, as for example, like 2.5D. There are again few ways how you can do it. You can merge projections, but in the easy case, you do this depth to color merge. So the idea is that you take a subvolume where you, let's say 3, 5, 7, 9 slices where you segment the middle slice only. So the amount of labor force work that you have to do is the same as doing 2D. And then you fuse this into the color channel image. And then after that, you take it to your lovely network architecture. So network, this way, it actually has information, a little bit more information about your structure, and which is especially useful for volume EM. And then you can basically use this in order to do the segmentation of the bigger volume. In this image, there's a segmentation of the A431 data set, FIPSAM data set. So Lena from our group, she segmented ER and mitochondria from 9 images, and then we were doing the segmentation of those. So here in the right bottom corner, you can see that there's a certain area which is this ER that was in the plane of, like in the cutting plane. So it was not properly identified by 2D network, but if you use 2.5D that has more information, it can actually be able to recognize that that part belongs to ER and not a lipid droplet, for example. So this is like a bigger data set that segmented multiple components, multiple organelles. This is also FIPSAM of U2OS cell. So you can see that this cell is very full with organelles. So there's lots of stuff. There are lots of mitochondria, Golgi, ER, and then naturally you can't really draw all of that. But now we can use deep learning in order to help us to segment all of this. Depending what kind of objects there are, some of those can be easily done manually because there are just few of them. But the bigger ones as Golgi stacks, mitochondria, or ER, we really have to go to deep learning because otherwise we can't really do it manually. There's no way. But the good thing was this, that on this quality of data and actually taking up all these new technologies about deep learning, we can actually get very nicely, very detailed, and quite precise models of these organelles. And then when we have them, what we do next? Oh, just one more. So in this case, we wanted to do it well. So we actually segmented 45 individual images, and then we did the segmentation. And if you want to do that, we have the data set is deposited to Empire. And also there is a segmentation instruction, which you can follow. They really describe how you can get from the data set to all these models. And there are also models that you can test and reuse and basically see how we did this. So, okay, we have the models, what to do with them. And this is a really big question because what we want to do eventually, we want to take these nice models and turn them into one plot with 2 bars, basically comparing your control with the treatment. So all that work basically for one single plot. And then this becomes like a problem for like, how we can get these 2 nice plots out of it? What kind of things we want to extract, can extract from this data? And then I would like a little bit talk now about this and then hoping maybe someone has a little bit better ideas. So in a very basic case, what we, of course, when we have everything segmented, what we can do, we can just calculate volumetric fractions. So okay, we know what would be the volume of the cell. We can know what was the volume of the nucleus in that part of the cell, and we can calculate the ratios. Okay, that's fine. But let's see about organelles. So with the organelles, it's pretty much the same sort of what we do. The easiest way we can analyze and compare conditions is by calculating what's the volume of our organelles in one case versus the other one, and basically scale it to the total volume of cytoplasm. Okay, but we have different objects. We have objects that are individual instances, like lipid droplets, like lysosomes, peroxisomes, sometimes mitochondria, or there are objects that form these big networks, like ER or Golgi. And then these, of course, require different types of analysis or quantification. If the individual instances as lipid droplets, of course, we can calculate their number, we calculate the average volume, average surface, dimensions. But what if it would be something more complex like ER? What we can do is this. So then we need to come up with our own metrics, perhaps, or try to find out what we can actually convert these models into numbers. One of the metrics that we've been using, I'll call it this kind of average minimal thickness. Like in the rough terms, what you do, you have a skeleton within your objects, and then you calculate the distance of pixels within the skeleton to the edge of your organelle. And that way you basically get the thickness of your structures. And this is applicable to all kinds of objects. And then we can use this in order to calculate, basically to get the distribution of thicknesses for the objects, and basically, again, compare treatments with the controls. Here's just some examples for mitochondria, for ER, or for nuclear envelope. Another example is like, okay, we have nucleus. So what we do with nucleus, we can actually detect the nuclei, nuclear pores. Well, in this case, we have a bit higher resolution data set. I just use morphological operations for that because I have plasma nuclear envelope membrane segmented. But of course, what we can do, we can actually do and just quite quickly just segment the nuclear pores through the whole nucleus. It's really easy. Just basically, you just need to click inside the center of, some part of this nucleus, click in the center of nuclear pores, approximate it as spheres, and then train a 3D network for that. Just really easy. But the results are really kind of nice, and then you can use this as the source for the additional analysis to get number of those, their distribution, their density, related to the total surface of the nuclear envelope, and so on. Another example, which is a very popular example, is like, because it's biology, so the organelles basically talk to each other, they form contacts. And what we need to do, we need to study the contacts. And what would be the way to study the contacts? In this case, what we here have, we used, again, this is like easy approach. You can use morphological operations, again, in order to find out the areas when ER touching mitochondria. And then you can use this in order to find out the contacts. We can find their numbers. You can calculate their average area, and then you can calculate related what would be the percentage of contacts that occupy mitochondria and those for ER. For example, in this particular case, mitochondria, 4% of mitochondria have contacts with ER. On the other hand, about 2% of ER have contacts with mitochondria. Okay, well, let's say, what about endoplasmic reticulum? So it's a complex network made of various morphological components, structures. In the simplistic case, we have sheets and tubules. So, okay, like what we can do with this, what we can actually try to split into tubes and sheets and compare the composition of the structure, because depending on the cell type, on cell cycle stage, you can have a different proportion between this. And you can always compare between the treatment condition. And in this case, you can see we use the, again, we have a tool to separate the tubes versus sheets, and then you can use that in order to calculate tubules to sheets ratio, and then potentially bring it up into comparison between your treatment versus control. Well, again, Elaine, it was a very nice talk about data deposition. I probably won't talk that much, and the time is ending, but I just want to say, you see there's one data set, but from one data set, we are able to extract multiple things. And then in that sense, it's important really because one particular research is focused on one specific scientific problem that I started in the beginning. So there are lots of things uncovered. In that sense, it really makes sense to deposit this to somewhere that people can use. And here I just put few possible repositories. So there's Empire for typically electron microscopy data by Image Archive, from light microscopy, and Zenoda for very generic stuff and the very kind of different types of workflows. Whatever you can actually put there. It's quite flexible, very nice system. So with this, I'd like to thank members of our group, Ilya, Helena, Elena, then also our collaborators from University of Helsinki, group of Elina Ikonen, they also Kirk, Lucy, Johanna, and Asta, who we together we had this paper on this kind of how we work with the data or how we do this reposition. Then Andreas Turtle from Zeiss for making one of these data sets, and also our funding agents and then communities. And thank you for your attention.
Great talk. Thank you. Are there any questions from the audience?
Thanks for the great talk. I was wondering for the microscopy image browser, it looks quite intuitive to use, so I'd like to try it, for instance, but are there also decent help files in allowing me to get a better grasp of what I'm selecting when I'm selecting for instance, let's say different workflows or different other subpanels?
You mean where in this deep learning?
No,
or
so in the initial deep learning part, for instance.
Yes. Well, we have the tutorials for that. Basically, there are maybe 4 or 5 different tutorials. There's one very recent one about 2.5D, but it's also applicable to 2D and 3D because it just brings you to idea about what you do, what you actually have to do in order to get to the results. Again, there are only a few parameters which are really important. The rest is like basically an optional things, which you can really benefit if you find out how to use them to your kind of, just to benefit the final kind of training, but it's not really needed. So in many cases, you really get to quite quickly to the results. Like if you're interested, I still like tomorrow, I can show you, we have here, we can do the full, like this data set of mitochondria, we can do in 3 or 5 minutes, the full data set of it, we can get the whole mitochondria from that. So it's very efficient, and you just basically just take 4 images, you segment anything to get this. It will take like 3 minutes, then you train for like 1 minute, and then you predict. It's a bit bin data set, but just to give you idea in the flavor.
Thank you for your interesting talk. Have you tested your 2.5D model on data where the pixel size are anisotropic?
Yes, so exactly. So the problem is, in a way, it's the best works when you have, it works on anisotropic, but of course, if there are big jumps, then of course it becomes less and less efficient. So the best way to utilize it more like when anisotropic voxels, but then if there's slight anisotropicity, it's still fine. So, but yeah, that's exactly the thing. So for example, if you have a data which is badly aligned, there are jumps and whatever jitter, you probably won't get good result with this. It probably will confuse the network more. So it's again, like you need a little bit know what you're doing in a way, but there are ways how you can improve the things sort of with the existing kind of methods that we have here.
Thank you. Quick question. When you begin to prepare your data for training like the 5 or 45 slices, how tolerant is it to error? Like I badly click or I forgot to click, you know?
Yeah, well, it's basically, you can, again, deep learning, it's statistical thing. So it's you have a lot of examples for the network, and it can't learn them all. What it learns is generic patterns, and it's more like statistical voting at the end, whether in most of your cases you had it drawn this way rather than the other way. That's my vision on this. I may be wrong. I'm not a kind of, computer scientist myself. I'm biologist, so I don't know. But I think it's just basically a statistical result at the end. The more examples you have like proper way, the sort of that what you result will get. But it's very important still to be consistent, because if you give it like plus minus quite big variation, probably results will be not as great. We've seen the examples of these approaches. It's better to have like a good models in the beginning.
So what you will say that you spend most of your time, how long it will take, for example, for your 45?
Yeah, well, we have exactly. So, well, it was before Segment Anything 2. With Segment Anything 2, it would be way quicker. So mitochondria, the easiest one. So probably took like there are 45 slices. It probably took like an hour to get this. Then Golgi was the second easiest one. It's probably was like probably half a day. And then the ER was the hardest because, again, what we try to do, we try to actually get it for the full slice. And then actually we have, I think we have 3 people sort of, or 2 or 3 people worked on that, and it was probably like maybe 2 or 3 days. So it's kind of really, but the ER is complex. You also have to like look a little bit back and forth in order to make sure they select ER, not something else. And there are really cases when you can't really distinguish between the ER or, again, there could be ER or parts of Golgi, so this becomes a bit complex.
Okay, thanks again for the great talk.